Have you ever wondered how DNA evidence is used? Well, they can compare the evidence of DNA at a crime scene with the DNA that is taken from different suspects. This lesson is on genetic technologies and engineering. So, one genetic technology that we see a lot in movies and TV shows is DNA fingerprinting. Now, this involves sending the DNA through an agar gel that results in a unique set of bands. And these bands are achieved because we've added a restriction enzyme that cuts the DNA into segments at certain points. And everybody's points are in slightly different locations. So, except for identical twins, nobody is going to have the same exact set of bands in a DNA fingerprinting test. So, this enables us to check for paternity or maternity, as well as to compare suspects against DNA samples that are found at crime scenes or even victims of crime scenes against known relative samples. Transgenics is a technology that takes genes from one species and transfers them into another species. And this is one way that we've created genetically modified organisms. So, this can include crop plants containing genes from other organisms that naturally produce their own pesticides to make those crops less likely to be eaten by um, herbivores. This can also be introducing genes into crops that make them resistant to chemicals such as Roundup. So we have Roundup Ready canola, where you can spray the herbicide all over the crop and the weeds will die, but the crop won't. This has also created mice that glow in the dark because they contain jellyfish genes to make a luminescent protein. The risk of transgenics is that we can produce unexpected effects on ecosystems if these organisms are released accidentally. So, again, that Roundup Ready canola has cross pollinated with weeds and other plants to produce herbicide tolerant weeds. And also, that Roundup Ready canola, if it gets into other crops or ditches, it's not able to be killed when you spray that crop with a herbicide because it's resistant to the effects of it. Also, transgenic organisms can produce bioweapons. So we could create a disease-causing organism that's designed to infect people or make them feel sick and possibly kill them. So if we created a vector that was able to deliver a disease to individuals. Scientists, though, are using the same processes in creating transgenic organisms to use gene therapy to help treat people who have genetic conditions. So, scientists can isolate a gene from a healthy individual to produce a substance lacking in other people, so like insulin in diabetes. This gene is then isolated and cut out of the healthy DNA. It will then be inserted into a plasma removed from a bacteria. And remember, those plasmids can travel into cells to introduce DNA. And this is one of the ways that bacteria share their DNA. So that DNA from two or more organisms is called recombinant DNA. So once then that recombinant DNA is inserted as an injection into individual's pancreas, those cells will then start to produce insulin. We also use gene therapy to treat cystic fibrosis as well as sickle cell anemia. So here for cystic fibrosis, they've taken healthy human cells. We've taken the healthy allele, inserted it into a plasmid in a bacteria, and then into an adenovirus that's good at entering human cells. When then we inject that into the individual suffering from cystic fibrosis, they're able to produce healthy lung tissue from those cells that have the healthy gene and the adenovirus in it. So we're going to replace the non-functional gene in an affected individual with the functional gene from a healthy individual. Now, currently, this is not a permanent solution, so people need to continue getting the injections for gene therapy because it doesn't replace the DNA in your entire body with the correct DNA, only in the cells where it would be necessary. So gene therapy can be used to treat conditions currently, such as insulin, people who don't produce insulin and diabetes, people who have abnormally shaped blood cells and sickle cell anemia, and people who have cystic fibrosis.